Hi, here comes lab video number 1. I'm not actually working the entire thing out. What I am going to do is open it and I am going to explain to you what you're supposed to do, just like if we were in lab. Then what I request is when you do the lab, hopefully you will have watched this first. Hopefully you will have your papers ready and then watch it again if needed. And as you try to go through it, when you come up with questions, my intention is you shoot me a message, you ask. If you have a really difficult time with this, then ask me about setting up a Zoom meeting and then we can do a Zoom conference and I can help walk you through it because some of these can be a little confusing. All right, here we go. This is what your page should look like. I have it as student view, so this is totally what you should be seeing. In order to get this video, you have already done your proof of attendance quiz, which means you're officially in the class. Yay! Okay, now, as I'm opening and adding more videos, I'm just putting them up here. Once I put them all up, then I will delete this part about I am making them throughout the week. I'm trying to get them all caught up by Tuesday afternoon. Trying very hard. Anyway, I am doing the lab video right now. So right now, you're going to go straight to lab. There is a lab safety video. The intention is you watch this video and then you go to the lab safety quiz and answer the question. Then there's the Embryology Lab and then there's the Speciation Lab. So I think I might just go ahead and try to do both of these real quick. So we're going to start with Embryology Lab. If you're doing the Embryology Lab, you click on this button. It brings you here. Notice it says, read the attached file, tell me everything, tell me what I stated in the instructions to tell me and convince me you watched the videos. So here we go. Click it, it opens it, well, downloads it. You open this file. Now, this is the cool part. So here you go, there is a game. You will click this button, it takes you to an actual game. Now, this, I'm hoping you can play this. Hoping. I love this game, but for some reason I have not been able to get it to play on my computer. You're supposed to click this Launch Interactive and then you see stuff. If you notice, I see nothing. This is what keeps happening to me. Some browsers work, some don't. Chrome does not work. I couldn't get Safari to work, and I don't think I got Firefox to work. I'm not sure. If you cannot open that, I understand. It's okay. If you can play it, then do this. Notice right here, I just said I tried it on 3 browsers. I couldn't get it to work, but if you can, it is interesting. If you cannot do it, then just go straight down here to the videos. This is one video, this is a second video. I want you to watch both of these videos. Then I want you to look at the comparisons of a human embryo, a pig embryo, a rabbit embryo, and chick embryo, turtle embryo, and a fish embryo. You can just Google these. You literally can just go, here we go, let's see, pig embryo, copy, and then open up a tab, click. This is a pig embryo. So this is what a pig embryo looks like. Now, let's see, we will do, I said chick, whoops, and then I start typing in something weird. I said chick embryo. This is a chick embryo. Yes, I clicked on images because here you go. So here's a chick embryo. Notice this will show you a whole bunch of different things. So basically I want you to look up the various embryos. I think, did I say turtle for one of them? Turtle embryo. Here's a turtle embryo. See, look at him. Isn't he cute? So this is what I want you to do. I want you to look at various, whoops, that's the wrong one. No, it's not. This is the right one. I want you to look at various embryos. And then what you're going to tell me is what you see that is similar about each of the embryos what stage they became clearly different from the human embryo. In order to do that, you can do things like human embryo, we'll say development stages. So here's your human embryo development stages. Here is something that would help you tremendously. Look at this. This one, wow, there we go. It's showing you a lizard, a chicken, a pig, and a human. So it's showing you some stuff. But you can do things like this. I want you to research it. I want you to look around. I want you to try to find various things because I want you to compare the different embryos to each other. So that is pretty much your entire lab for this embryology lab. Now, I used to have more to this, but Flash is getting disabled, or it's going bye-bye, and some of the sites that I used to use have stopped working. So we are, you guys are number one of me trying to bring in new stuff. So this is where it is. So your main thing for this one is watch the stuff, tell me about the videos, and then compare all of these. So all of those embryos, and tell me what's similar and when they kind of deviate. So basically, like, is it really close for the fish and the human, or are they very different? Does the fish and, say, the pig, are they very similar for a long time and then they change, but a human and a chick are alike here and then they change really quickly? That kind of thing. Figure out the best way you want to word it. I just want to have proof you watched both videos and that you researched this. Showing me pictures or telling me where you went, giving me hyperlinks or, websites. That would be great. I just want detail. So prove to me you did this. All right. Now we go back to week 1 and we talk about the Speciation Lab. Speciation Lab can be a little crazy. Now, this is where you're going to turn it in. So when I click on this button, notice it's just one of these. So you would then click Add Submission. Make sure you click this little thing, and then you can type words here, and then you can attach your document here. When I say attach your document, make sure it is a Word document, as in it ends with .doc or .docx. Or you can attach a PDF. Those are the only two that tend to properly open through Moodle. If you attach it in a format that I cannot read, I cannot grade it. So make darn sure. After you submit it, go back and double check. If it's just messed up, that means I can't see it. Send me a message, we'll figure something out. Now, for the Speciation Lab, you are going to go here to this folder and click it. Notice there are 4 things here. You need all 4 things. You can either say download folder, it will give it to a zip file and you open each one, or you can just do this and click on every single one. Either way, it works. Now, I am going to show you, this is a grid. You will want to print this out because this is what you're going to be doing part of the lab on in order to answer the directions. This is all about the lizards, the salamanders that we're covering. So this will give you some information and tells you which ones they are. Then we have the questions and we have the information, the instructions. Now, you need all 4 of these files to do this lab. What you will do is start with instructions. So here it's telling you all about this California salamander. This is kind of important. These are words and these are going to help you answer some questions. So you kind of want to know this. Here it's telling you what you need, your materials. You need colored pencils or crayons or markers. You need something that you can clearly mark on that map. I would recommend when you print the map to print it kind of light, because you see it's very, very dark. If you print it lighter than that, you will be able to see what you draw on there. Much, 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 much better. Okay, so print off the grid map of California. Then print off your subspecies of the salamander. You don't have to print that, but it kind of helps you. Now, begin by reading the background information below, that's coming down here, before you go towards the exercise. Now, a lot of people really like to just jump and try to answer the questions. If you don't do it properly, it's going to make it harder. Then at the end of the lab, you're going to turn in your colored map and your questions for grading. This is going to be through Moodle, okay? There's a part where I'm going to ask you to take a picture of yourself holding the map so I can see it. And so I can see that you yourself did it and you didn't just copy it from someone else. You have to do this. It does not mean just scan in a picture of the map. I have to see you doing it, okay? Now here's the background, and so it's talking about these salamanders. Notice this is highlighted and says must start with this. If you do not start with part A, you can't do part A. Notice the questions where it says part A based on part A of the lab. It means you have to do this part A. When you are completed, then you can answer these questions. If you don't go in that order, you are not going to be able to answer the questions. Now, this is telling you all about the different salamanders. This is explaining that each one of the salamanders is giving you information. So it is telling you, like here where it says E, and then it says 230, and then it says yellow. This is telling you what color you're supposed to be coloring your grid, your California map. If you're coloring it yellow like it tells you to, you're going to know that this salamander lives in that area and it was a population of 230. So that's how that part works. Now, here, when you see these, this is what matches up. So A means that's the name of the salamander. There are 15 of these guys in this location. You are to use the color brown on your map. Now here's the big part where it says 32R. 32R means you are going to look here. You are going to find the number 32 right here. I wish I could highlight it with this. But anyway, you will find the number 32 and the letter R, and you go up the R until it matches the number 32, and so this is where you're going to mark. So you will mark this. Then the next one is 32S. So you now go over to 32S. Then you go to 30T and 31T. This is basically data point, data point, data point, data point, connect the dots, color it in. That is the area for where that salamander lives. You're going to do this for each of these salamanders. Notice this one tells you color it brown, this one's red, this one's blue, purple, yellow, green, and orange. Can you use different colors? Sure. But if you do, you need to make note that you did use a different color, because everything is color-coded to help you identify something. Now, notice I put something in red. What I put in red is, this is one of the biggest mistakes people make. Notice this is a capital R, this is a capital W, this is a capital B, this is a capital C, but this is a lowercase a. It's not a mistake. It is not a typo. This is intentional, because if you look on this map, These are all capital letters. And then we have a lowercase a, b, c, d, e, f, g, h, i, j. All these are lowercase. Why this is important is because if you say that this salamander is living in 40A and you don't use this A, it's telling you that this salamander lives right here. But if you accidentally use the capital A, you have just stuck this poor salamander out in the Pacific Ocean, and he can't live there. So you have to be very, very, very careful. So be careful, blah, blah. Why do I keep doing that? So be very careful on exactly where you mark these. Now, plot each collection area by filling in the corresponding squares, like I just said. Data points, connect, color. Take a picture of yourself holding the map that you just plotted. And then answer the questions for part A. So now that you have finished part A on the grid, you will then use that information to answer these 5 questions right here. Then notice this part says based on part B. You can't answer it until you do part B, which should be, oh yeah, it's right there. I just kept seeing this and was missing it. This is part B. You have to do this after part A. If you do it before, or if you just aren't paying attention, you can't answer any questions. So now that you have done part A, you are going to do these. Notice you have different colors than in part A. This is so you can compare part A to part B. So again, notice you have the spots that you mark. So like 36Z, you would go to Z and you would go up to 36, which is right around here. And then that is where you put the mark. Okay, once you're done with all that, mark with the color gold these places where they searched for them without success. Then notice specimens of population 8 and 9 are shown in the color pictures, but there are no illustrations for populations 11 and 12. That just means they're not showing you on this. They're just not there. So once you have done this, you then come here and you answer the questions for part B. Do your best. Try to look at it, try to understand, pay attention, and go back to looking at some of the stuff that is covered in the lectures for this week. We are doing chapters, 12, 13, and 14, I believe, this week. Again, brand new quarter. I'm still learning because it's also a brand new textbook. So let's see. We are doing, yes, 13, 14, and 15. Those are the chapters we're doing this week. Make sure that you use your textbook to help you answer some of these questions, because it's going to ask you a little bit about speciation. It's going to ask you about allopatric speciation. It's going to ask you very specific things, which will be covered in your textbook and in the lecture video that will be posted very, very soon. I say that because if you happen to open the lab before I get the lecture video recorded and up, then it's soon. If you are late starting for the week, then, the lecture video might already be there. But you have to actually prove to me you have done everything. Take your time. The Speciation Lab can be very frustrating for some people. Other people don't have so much trouble. I can promise you, if you actually follow directions and you go in the order I tell you to, it's not that bad. If you try to just skim it and jump to it and you're trying to shortcut it, it's going to drive you crazy and you won't do it right. Just so you know. Yeah, that's it. Those are our labs for the week. So make sure to get them done and turn them in the appropriate places. The lab safety quiz is right there. It's titled Lab Safety Quiz after you watch the video. Embryology Lab, Again, you click there, it's got the document, it tells you what to try to do. It's going to take time because I expect you to actually look up the various embryologies here. So human embryo, pig embryo, rabbit embryo, and whatever other embryos I told you to. And then Speciation Lab, 4 documents here, 4 files. You need them. You have to print out the California grid. You have to color it. You have to mark it up, and you have to take pictures of yourself holding it, and all of those have to be turned in. If you do not know how to turn in a picture, I normally tell people to open a Word document and just insert picture. You just put the picture right there in the Word document, and then you can type up all your answers. It makes a nice, neat one file. If you have trouble with this, contact me, let me know, and I will do my best to meet up with you, meaning via Zoom, to help walk you through it. Because if you want to do a Zoom meeting, then I can set it up and I can show you my screen and I can totally walk you through it 100% and teach you how to do this, and it will help you tremendously. I think that's pretty much it. So yeah, those are the labs for week 1. Hopefully you survived. Yay. Now I just have to make the lecture videos and tell you what your lecture assignment is. So I guess that's it. Hopefully everybody's okay for week 1, because whew, what a way to start, right? All right.